Welcome everyone to the CGIL seminar. Today we are delighted to have with us Lucas Alcantara. He's a student here at the University of Guelph, and today he's going to share some of the things he has done throughout his PhD. So after his presentation, you are going to be able to unmute yourself and interact with Lucas if you have questions or comments. And for that, you can also use the chat box on Teams. So without further ado, let me introduce our speaker. Lucas obtained his bachelor's in biotechnology and a master's in bioscience, both from the Federal University of Bahia, Brazil. As a master's student, he studied the role of different bacteria from the rumen in the degradation of plant cell wall through bioinformatics analysis, aiming for species with the potential to produce enzymes of industrial interest. Lucas is currently a PhD candidate from CGIL under the supervision of Dr. Flavishenko and Dr. Christine Bays, and he has been leveraging data-driven algorithms to provide information and solutions for different areas in the dairy industry. This week, he will talk about his work to provide a methodology to develop classification models aiming to identify text-based breeding protocol descriptions of hormonal synchronization protocols and how this will help researchers to further understand the effects of timed AI on genetic evaluations for fertility traits. In addition to his PhD studies, he is currently working as a part-time at the Amafra Elora Ponsonby Livestock Research Center as a livestock data technician. So I'll pass it over to you, Lucas. Thank you.
All right. Thank you, Ivan, for the kind introduction. Let me start sharing this. Okay. So as Ivan mentioned, the title for our presentation today is Building a Machine Learning Model to Identify Text-Based Descriptions of Hormonal Synchronization Protocols. I would like to keep this presentation a bit informal, and in the end, please, you can join me on questions if you have so. A bit of a summary of what we are going to go through today. I'll describe our problem, go through the methodology results, discuss them a little bit, and go through some final remarks. Okay, so timed artificial insemination protocols has proven to be a successful management tool in dairy cattle because it affects the reproductive physiology and endocrinology involved in the estrous cycle, thus allowing for higher conception rates even for genetically inferior animals in terms of fertility. However, timed AI is known to mask an animal's true fertility performance, which reduces the accuracy of genetic evaluations for fertility traits, as recently shown by Oliveira and Lynch in the last couple of years. One of the suggestions proposed by Lynch to address this possible bias was to split fertility traits by the management technique involved in the breedings. The goal with this is to have unbiased genetic evaluation of animals according to their true fertility performance. But this task is rather difficult because there is a lack of specificity and uniformity in the recording of breeding protocol descriptions by dairy farmers. This makes it very hard to identify which protocol descriptions are from timed AI protocols and which are not. But what are these protocol descriptions anyways? When an animal is bred, farmers indicate on dairy comp the breeding protocol code used on that service. Each code is a letter that is linked to a short description of that breeding protocol, which is normally a combination of only a few keywords, abbreviations, numbers, and or special characters, such as we see in the list, natural, off-sync, standing, GG, PG, and cider. However, because there is no standard for creating these descriptions, two farmers might use different descriptions for the same or variations of the same protocol. For instance, Lynch reported that there were 156 unique descriptions describing off-sync in their dataset as you can see at the bottom here. And the source of the variation came from typos, capitalization, and or abbreviations. Another factor that makes working with breeding data challenging is that farmers create some descriptions that are very much not clear, or for protocols that are hormonal treatments, not necessarily for timed AI, but for sick animals. So the database linked used from Canadian farms contain almost 6,000 different breeding protocols descriptions across around 1,000 herds, of which 2,021 of these appeared in only one herd. So, but how do we deal with all these different descriptions? So let me introduce an approach from the era of natural language processing known as bag of words representation. This form of representation associates a text with a vector, indicating the number of occurrences of words from a predefined dictionary. So in a list of sentences, as we can see here, let me put my laser pointer. So a list of sentences becomes a sparse matrix of frequency counts. In this example here, the words in the header are unique words taken from the list of texts, and this is what we call a dictionary. For the first text, the red dog, we would count how many times the words in the predefined dictionary appears in the text. In this case, instead of using the text as the feature, we would represent it as a vector within with the frequency counts. So instead of using the red dog, we would use 111,000. But if you remember, breeding protocol descriptions are normally short descriptions. They're not a full sentence like this. So the same word would be spelled differently as well. So we would have slight changes to represent a change in a protocol. So using full words like this is not the best approach for our problem. What we could do is to adapt this approach by using combinations of character bigrams instead. Again, we would construct the dictionary from all possible 2 consecutive characters in the text list. So CA, AT, DO, OG, and so on and so forth, and vectorize them according to the bigrams in this dictionary. We will then create a matrix of frequency counts by putting all these vectors together. So now you have a better idea of what the problem is and why and how to convert these descriptions into vectors so we can train our models. So let's take a look at what we did in this work. So our objective was to build robust machine learning models able to identify text-based descriptions of hormonal synchronization protocols. I would like to spend a little bit of time in the methodology because I think it will help us understand better what, why, and how we did things the way we did, as it's a little bit different than traditional machine learning approach. So let's go through our data. The data we used was recorded by Canadian hosting farmers on DairyComp from 2009, 2007 to 2019. This data was kindly provided by Lactanet, and it contained around 800,000 cows from around 1,000 herds. This is the very same data set that Lynch used in his previous work. From that data, we removed herds that did not have any breeding protocol descriptions on the program, and we ended up with around 700 cows from around 1,000 herds. And this is pretty representative of what we have in our national herd, which is about 70% of hostings in Canada, according to the census in 2020. In this data, we found around 6,000 unique breeding protocol descriptions, and it varied from 1 to 24 descriptions recorded per herd, and a herd on average had around 6 descriptions. We then grouped these breeding protocol descriptions into 2 main classes, timed AI and not timed AI, so other. For timed AI, we said it was timed AI when timed AI protocols was used alone, like just off-sync, or in combination with any other protocol like PRID and estimate or off-sync and natural. So if there's any trace of timed AI, clear trace of timed AI, it would go on group number 1. On the other hand, when breeding protocol descriptions did not indicate any use of timed AI protocols, such as hit detection, hormonal use, and unclear descriptions like vet advice or dead set or just numbers, they would all go into other. So this approach is so we can have a clear timed AI group against everything else. And one assumption from this work is that these classes, they were previously labeled manually by Lynch, and they are highly accurate. The first thing we did with these descriptions was to convert uppercase to lowercase and underscore to white spaces. This approach alone reduced the number of unique breeding protocol descriptions by 26%. Next, we look at the number of new unique breeding protocol descriptions added to the dataset every year. We can see that the number of new codes being added stabilized after 2010. And we also noticed that there was an average addition of 140 new unique descriptions every year, starting in 2011. And this represents around 16% increase per year. So we understood in that moment, when we looked at this graph, that we needed to build a model that would be robust enough to maintain a high performance, even with this number of unseen data being added every year. In other words, we would need a model that did not require frequent training, that could handle new data being added every year. So to test that, we had to come up with an approach that would mimic what would happen in the future. That is, train the data with only a certain amount of data up until a certain year and test individually on the following years. Our splitting strategy is very different than the textbook approach of 70-30, 80-20 split. So we actually split the data according to the year of insemination. We use the full data and split it into 9 training data sets that were grouped by the year of insemination. As you can see on this table, they were incremented yearly. So we had 9 of them, and these are the number of records per training data set. Okay, so remember the character bigram that we talked about? So this is where it comes to play. We extracted a unique dictionary containing the combinations of every 2 consecutive ASCII character in each description. And this was done for each of those 9 training data sets. Then we used a vectorizer function to convert breeding protocol descriptions to row vectors containing frequency counts of bigrams from said dictionary that are present in each respective breeding protocol description. So we went from a data that had 2 columns, description and the class, to 9 sparse matrices, which were then used for the training and of these models. So here we can see the number of features that each of those training data sets had. And we see it ranged from 491 to 727 bigrams, which represents an average yearly increase of 4.5% in the number of features. As for the testing data sets, we created 45 of them, each consisting of unique descriptions used in the years following a given training data set. So for example, the training data that had all descriptions from examinations performed from 2010 to 2007 to 2010 was tested 9 times, one for each year that followed that data set. Whereas the training data from the training data from 2007 to 2018 was only tested with new unique descriptions added in 2019. But there is a catch, because when you train a model with a certain features and a certain number of features, the test set must have the same features. So instead of building a new dictionary for each of these test sets, we used the dictionary from their respective training set when we built the matrices for the test sets. As you might be imagining, all test sets from 2019, for example, they were completely different from each other. So this one had 491 features, this one had 540, even though they were from the same year. So as you saw a few slides ago, the number of new descriptions varied across years. So this would not be very fair to compare models that had different number of records in the test sets. So we picked a year that had the less number of records, which was 2019 with 87, and we made sure that all 45 testing sets had 87 randomly sampled records. So here's an overview of the modeling pipeline that we used. We ran this 8 times, one for each algorithm. Each algorithm had their hybrid parameters optimized for each training dataset, which then produced 9 optimized models. Each of these models, optimized models, were trained on their respective training dataset. They were evaluated based on their training and validation performances. And we checked for over and underfitting of these models based on their performances. Lastly, each trained model was tested on their respective training datasets, which were the ones I showed you in the previous slide. But I want to bring your attention to this algorithm here called stacking classifier. It goes a little bit different than this pipeline. So we would have each of these algorithms trained, optimized, and trained, and then we would end up with 9 models per algorithm. What we would do is, for a stacking classifier, let's say that the trained model N, maybe the model 1 or 2 up until 9. So this is the model that was trained, optimized, and trained for K and N, for the K nearest neighbor. This model had its predictions. Same for all the others. The predictions produced with part of the training set, the respective training set, they were used by a meta classifier to understand its predictions and make a final prediction, taking all of those into consideration. As a meta classifier, we used linear regression, and we didn't optimize any of this because we already used optimized models here. So we didn't want to further fine-tune this model. But this is the gist of what a stacking classifier would be. So we would optimize all of these and use the optimized trained models to build the stacking classifier and get our final prediction. And then this stacking classifier would then be tested on all those training sets here, just like the other ones. Here are the performance measures that we used. I'm not going to go into details into those, but We used Matthew's correlation coefficient as our main performance, but we also use precision and recall to carefully interpret those performances. We also wanted to maximize precision and recall. Thus, we looked at F1 scores for that. So as MCC was our main performance, whenever I say performance or score, I will be mainly referring to this coefficient unless I tell you explicitly. So next thing we did was, if we dive into that pipeline, we optimized model hyperparameters. And for those that aren't familiar with a hyperparameter is, they are external user tuned parameters. And those values, they control various aspects of the learning process, such speed and quality, like the tree depth in decision tree models or number of neighbors in KNN models. And these parameters, they cannot be inferred while you fit the model to the data. So they are user defined. They're external before you fit the data. So to optimize, we used a Bayesian search approach to produce various hyperparameter combinations from the search space. For each combination, we use a repeated stratified tenfold cross-validation to evaluate the performance of a model using search combinations of hyperparameters that were produced by the Bayesian search. And the set of hyperparameters that produced the maximum MCC scores in the validation, the holdout data, was ultimately selected for any given model. As an example here, we have the hyperparameters in the search space used for KNN. So we optimize the number of neighbors, the leaf size, and the distance metric. So we went from 1 to 11 every 2, from 10 to 50 every 10, and we use these 6 metrics for distance. So for both optimization and training, we used a repeated stratified tenfold cross-validation. If you're not familiar with how this cross-validation works, briefly, each data set being evaluated was split into 10 parts with a similar distribution of records per class. That's what the stratify would mean. And in this case, we had 70% timed AI and 30% other. So one part was set aside, that's what we call the holdout for validation purposes, while the other 9 were used to train the model and measure its performance. This process was repeated 10 times until all parts had their turn to be used for validation. And then once I finished this, I would repeat it 2 more times. So that's why it's repeated. So we did it 3 times in total. For the optimization, this was performed with all different combinations of the hyperparameters from the search space that was defined by the Bayes search. And for the training, it was done with the then optimized model coming from the optimization. So performance on the validation parts were averaged, and I will refer to them here as validation scores, while the average performances of the model on the other parts that were used to train the model, I'll refer to them as training scores. All right, so let's look at some of our results. In this figure, we have the training results of all algorithms. Let's focus on the decision tree for a second so I can explain the plot. On the x-axis, we have the training sets. And on the y-axis, we have the MCC scores from the cross-validations. The orange box plots are the validation scores, and the blue, we cannot even see the blue, but they are the training scores. So going back to the full plot now, most algorithms had very similar and high training and validation scores, as you can see here. What I want to highlight is that stacked had the best performance among all algorithms. It had the highest MCC on all validation data of all training sets, being the one with data from 2007 to 2015, which would be this one here, to have the highest validation score. On the other hand, linear discriminant analysis had the lowest performance among all. It had the lowest MCC on all validation data on all training sets. Still, its highest performance was achieved when data was used from 2007 to 2017. So let's check now the results from the test sets. In this table, we have the average results of the various models that were fit on the test sets. For example, the last row here represents the average result of the models trained with data from 2007 to 2010, from 2007 to 2011, to 2012, 13, 14, and so on into 2018. On the other hand, the top row is the only one that is not an average, since only models trained with data from 2007 to 2010 were tested on data from 2011. Looking at the last column here on the right, we see that models built with the stacked algorithm outperformed all other models when tested on unseen data. They also had similar high MCC scores on testing data, which had an average of 0.94. Even though stacked outperformed all others, they all have high performances, except for KNN and LDA. This shows us that no matter how much data I include in my training set, I get similar and high performances. This is exactly what we were looking for, an algorithm that could be used to build a model with data from today that will have a performance in 10 years from now as high as it had today. So there's no need to optimize and retrain this model yearly before every genetic evaluation. So for the sake of ranking these algorithms, let's go back to our objective. We want to identify as much timed AI as we can, as correctly as we can. We can do that by looking for high F1 scores, which means maximizing precision and recall. So let's get these measures understood better for our problem. So if we have a precision of 1, it means that all timed AI observations were correctly classified as tie. On the other hand, if that precision is 0, all timed AI observations were wrongly classified as other. So we messed it up. We didn't get them as correct as possible. For recall, if it's equal to 1, all observations classified as timed AI are truly timed AI. On the other hand, if it's 0, none of the true timed AI are classified as timed AI. So in this case, we wouldn't find any timed AI in the true data set. And in this case, we would find all timed AI that were there to be found. So we want to find a balance between these two. But really, the best scenario is if we had an F1 score of 1, which means I would have a precision of 1 and a recall of 1. That's the impossible goal. We always have the irreducible error, but that's what we aim for, to maximize precision and recall. So the higher the F1 score for us, the better. Like the previous plot, we have here the training sets on the x-axis, but now on the y-axis, we have F1 scores. Values on the box plots here, they represent the results for all test sets obtained with a model trained with that particular training set. So The last result here from 2007 to 2018 is actually a flat, solid line because it was only tested on the 2019 data. As for this one, it was tested on all of the 9 test sets. As you can see in this, there isn't much fluctuation on this. So incremental addition of new data to the training set did not help the maximization of F1 scores for all models, except for LDA that we can see it's getting up. We are maximizing, we are increasing F1 score as we add more. This results reinforces the MCC results that the machine learning algorithms used in this study, except for LDA, they are robust and can be used to produce models with consistent high F1 scores, regardless of the amount of data you use for them. If we average all results in the previous plot by algorithm, we get this table. And we can see that stacked had the highest average F1 score. However, results aren't very different among algorithms, which gives us a better flexibility to choose a preferred algorithm for the job. If we want to go for interpretation, we might as well go to decision trees, which are super simple to interpret. Whereas for stacked or random forest, not so much. So this brings us to our final remarks. So let's recap a little bit what we did. So we wanted to classify breeding protocol descriptions into timed AI or not timed AI, what we called other. We constructed features representing the combination of 2 consecutive ASCII characters. We optimized, trained, and tested models built with 8 different supervised machine learning algorithms. And here's what we found. We found that stacked showed higher performance, MCC of 0.95, on average in each year that followed the years used in all training data sets. We also found that precision and recall were maximized by stacked models with an F1 score, with an average F1 score of 0.96. And we can get from these that stacked is a strong candidate to produce machine learning based models to correctly identify timed AI protocols. And now we know that with this study that machine learning algorithms can be used to produce robust models that correctly identify timed AI protocols from dairy cattle breeding records. This is important because it opens the possibility for unbiased genetic evaluation of animals based on their natural fertility. And I would also like to add that it will help researchers because now they can know what breeding protocol was used for each of those inseminations, and they can further study fertility and understand more the effects of timed AI. With that, I would like to acknowledge our funding agencies that were involved in my PhD so far, and I am happy to take any questions you may have. So thank you very much.
Thank you, Lucas. And I will make it possible for you in the audience to interact with Lucas. Just a moment. I allow. All right. So now everybody has the opportunity to talk with Lucas. So if you have a question or a comment, please raise your hand and I will call you. And yeah, you can talk to Lucas. You can unmute yourself and open your webcam as well. All right, I see. Let me see. Well, the first hand I see here is
I believe it's
Dr.
from Dan.
Topin.
Yep.
Yeah, so please go ahead.
Hi, Lucas. Really nice presentation, really clear.
Thank you.
I just have, I want to clarify something quickly. I think on one of your slides, slide 23 or something, when you showed the pipeline, I think you mentioned that you use linear regression as a like a general, like the meta classifier. Are you
Yep.
sure it wasn't logistic?
Correct.
Okay, just to make sure, okay, that everybody else understood.
Logistic regression. Thanks, Dan, for the
Okay, no,
reminder.
just that. Okay, thanks.
All right, and then Flavio, please go ahead.
Great presentation, Lucas.
Thanks, Flavio.
Difficult topic to present, but we made it very, very easy, very straightforward. I have a question for you that I started thinking during your presentation. When you are showing the strategy to optimize the hyperparameters,
Yep.
then you end up using the average of the final MCC.
Yep.
So I wonder if there was really a clear difference in averages that you could say that is the best combination of hyperparameters, or if there was a situation that you have like similar averages that would be worthwhile to look at the standard deviation, because you could have a very similar averages, but right, one combination of hyperparameters to have a more fluctuation with a higher standard deviation. So that's a situation that you don't like.
Yep.
Right? You like to have a high average and a very little variation.
That's a great
It
question.
shows that more robust the combination.
Yeah. I can tell you right away that yes, they were very similar. So sometimes when I was writing the code, I could see that, for example, for running KNN, I would get sometimes Manhattan, sometimes, you know, uh, Hamming distance interchangeably and the same results, right? So I think models, they would really get similar averages, but I would have to look back into the implementation of the software if there is any account for standard deviation when they suggest the best parameters. So, but I do agree that is an interesting observation. But regardless of that, Flavio, when we get the parameters that we ended up using, in the end of the day, the training, they all had very, very small standard deviation. So would it be worthwhile to keep tuning to go from 0.04 to 0.02? Maybe, or maybe not. But that's a good question, and it would be interesting to look at.
Yeah, maybe not for your results, right?
Yeah.
As you said, there was very little. The standard errors are very low, right?
Yeah.
But in other situations could be important, right? Because
Yeah.
you really could have similar average, but quite different fluctuation across
The standard deviation.
deviations, right? So then it might be important to look at standard deviations too.
Yep.
I was just curious if you look
I
at it.
agree. I haven't looked at that, but I agree fully. Yep.
Okay, thank you, Lucas.
Thanks for the question,
Great
Flavio.
presentation.
All right, I see Lucas Lopez have a question, so Lucas, go ahead, please.
Yeah, there we are. Thanks, Lucas. Great presentation. I think this is the third, fourth time I see this presentation, and it's getting better and better. So congratulations. I think
Thanks.
I was finally able to understand the complexity of the project you were involved in and the problem that you had to solve. So it's very, very nice. This is more of a curiosity question though. Working with this kind of data coming from, real environments, what was your what was the most difficult and hard thing to deal with going through it, if you would pick one?
Well, I could pick lots of them, but
I figured.
think, yeah, I think the most challenging part is because when we get data that comes from human input,
Mhm.
there's always a challenge of getting clear data and matching like when I was looking at the statistics and what records were there, what weren't, it was always hard to check because they would always have something missing, something not matching. In this particular case, it wasn't very hard because when this data was extracted from dairy comp, it came for, this is what Colin worked with. It came a record of bread events, and those records had the letters that were corresponding to the descriptions. So working with that bread event is really chaotic.
Mhm.
But for me, I only used the second file, which has the descriptions. So it was much clearer to work with. But when we decided to go forward and split by year of insemination to see when when those breeding descriptions started to appear in the data set, that became a little bit challenging because we would have to go through the dates there that weren't always the same. So for example, we had data from English and from French. So sometimes the year and month would be different. So I would read the data and then, no, this doesn't make sense. Oh, it's because the date's different. So those were some sort of challenges with this, but I think the most challenging was how to build this training and testing data sets and how to organize them all and loop through all of them in the program. But I've worked with more challenging data sets, that's for sure. But this one was okay.
Okay, thanks, man. Congratulations again. Really, really nice.
No worries. Thank you.
Okay, we have another question from Don. So please go ahead.
Saving hardest for last, Dan.
Sorry, Lucas. No, I don't want to put you on the spot or anything like this. It's just out of curiosity. Since now your problem it's a binary problem, so I think originally, at the beginning we looked at a 3-class instead of a 2-class problem. There are certain train of thoughts and people of the opinion that maybe an AUC kind of analysis might make more sense to compare the 8 algorithms, and there are pros and cons on what or how to use it and why to use it or not use it. With an alternative, there is also an alternative. Since you have already computed precision and recall, and you group them through the harmonic mean via the F1 score.
Yeah.
Sometimes the F1 score can hide certain things, right? So especially if you have more interest into one versus the other, so it kind of balances them with equal weight. But sometimes you don't want that. So as a suggestion more than a question, or maybe if you want, I can ask it as a question. If you ever considered to use maybe a PR curve, so precision recall curve, to basically rank the performance of the algorithms.
Yep, I looked into that, but I think I did. Well, I didn't go with it because for this specific problem, I do want to have both precision and recall maximized. So I want to find as much as I can to make sure there's no timed AI mixed in with the genetic evaluation data set. And I want to make sure that I am really finding, like if I'm telling it's timed AI, it's timed AI. And the other way around. So both of them are very important. So that's why I didn't go to a precision recall curve. So But that certainly is, there certainly is value on looking on those separately for other kind of problems.
Sounds good.
All right. Does anyone else have a question for Lucas? Well, I have a question, Lucas. When is your PhD defense?
Well,
I know
as
it's
you're going to be my
approaching.
student representative, it
Yeah, remind me, please.
it's going to be on, I don't know if you can confirm that, Flavio, but it's June 14th from 9 to 12:30. Can we hit a hammer on it, Flavio?
Not yet. That's the plan, but it all depends on the
On the
yeah.
examiners?
We have to formalize. No, the examiners are
Okay.
fine, but we need to formalize the request, and
Okay.
until the graduate studies don't agree with it,
Okay.
but that's the plan.
Okay. Okay.
And probably, Lucas, will be from 9:30 to
9:30.
12:30. Yeah.
Okay.
But
All right. So it's that week, Ivan.
Okay. Yeah.
Save the week.
No, I think the day will be May 14th. That's
yeah. June
only,
14th.
June.
Don't kill me.
sorry, June. Ernie
No.
Master is defending on 16th of May. So
Yeah.
sorry.
Yeah.
June 14th.
Yes.
Yeah. So this would be my second study on my PhD. So yeah, it'll be nice. This was a nice warm up for the day.
Right. And do we have another hand up? It's actually, yeah, Dan.
Yes,
Three
so
times
it's
a
me
charm.
again. I just wanted to finish a question also about the presentation. Now that it's all clear with the thesis when it is, right? So the defense, I mean, it's I want you to speculate on something, and I want to see what's your opinion about it. So let's say you want to take your model and apply it on a similar data set that comes from a different country that doesn't speak English.
Okay.
But they use the same alphabet, the same characters. No,
Yep.
Nothing else, right? Do you think it's going to work or not?
Oh, 100% will. 100% will work. This is one of the advantages. Thanks for asking, because I didn't touch on that. So we wanted this to be flexible. So it doesn't. We are not training on specific words. We are training on the relationship that those or any combination of two characters will have with our targets. So if it's English, if it's French, Portuguese, it will follow the same pattern. Obviously, I will have to train it on that language, but that should obviously work just fine. The same methodology.
Okay, so actually, yeah, you answered a slightly different question, which is more generic. I like that. So you still have to retrain your model. You cannot use the same model. That's what I meant.
I would expect that I would have to retrain for a different language, because it will be a different dictionary, like completely different.
Okay, and what do you think is going to influence the performance of your model if you train it, let's say, on a different language that uses the same alphabet?
Well, for example, if we think that if we go for natural heat, if I say in English, or let's go for heat, I think that's a better one. Heat in English, heat. But in French, I don't know how to pronounce it. I'm sorry, Audrey, please don't kill me. But I know it's CHAT.
Chaleur.
Yep. So it's completely different. So the letters in the word will be completely different. So it might be a little bit hard to have just English or just French there. If we have a bit of both, they may pick up on on the records and recognize that both natural, on both heat and both that one will be for will not be timed AI. So if the languages are similar enough, okay, but if not, I don't think so.
Okay, so do you see a perspective where you might be able, let's say in a not so near future, to collect data, let's say from maybe 10 or 15 countries, similar data like this, and train a multilingual model instead of just this? And how do you perceive it's going to perform? Same performance, lower, higher?
Oh, this would be interesting. I haven't thought about that. Nice.
Well, I know you're going to have a lot of time on your hands after you graduate, so you know.
I think that would be nice. That's for sure. Yeah. I'm just concerned about too much variation in there. Like, for example, OV may be very, very frequent for OVSync, but maybe some different language would have millions of records of OV that is completely not related with OVSync, and that will confuse the model. So there will have to be some tweaking in there to figure out a way. I don't know, maybe put like a language variable there and to help the model say, oh, it's OV. If it's English, okay, timed AI. OV if it's whatever that language, no, it's not timed AI. So that might help with the process.
Pretty cool. No further questions, I promise. Thank you.
Save them for the defense. That was actually a great question, then. Thanks. I haven't thought about that.
Yes. Well, Lucas, I think you had a good opportunity to test your knowledge on what will be soon your defense, right?
Yep.
So I want to thank you for being part of the CGIL seminar. Today, this is our last seminar, so we conclude the series of the winter semester, and we hopefully will be back in the fall. So we have now a summer break, and in the fall, we'll be back with another series of CGIL seminar. So thank you, Lucas, and thank you all for participating, joining us during all this semester. When we are close to, let's say, September, probably around that time, we will be sending out emails with a list of speakers for the fall semester, and I hope to see you back again in September. So thank you very much. Enjoy your weekend.
Thank you very much, Ivan and Colin, for organizing all this. Oh, it seems I'm reading Christine's. Yeah. Thanks a lot. It was great.
Thank you, Ivan.
Thanks very much.
For organizing, Colin. Well done. Great semester of seminars. And thank you, Lucas, for being the last one.
Yeah, today. It was great. It was very nice. Thanks.
Great job. Thank you.
Okay.